We will talk about endoplasmic reticulum, shortly called ER, and it is a cell organelle found in eukaryotic cells. These are found in eukaryotic cells. In case of bacterial cells, prokaryotic cells, these are absent. Now, endoplasmic reticulum was first discovered by a scientist Porter in 1953. Some of our cells like pancreas, etc., the quantity of endoplasmic reticulum is higher. It is usually higher in those cells which take the function of protein formation. It is called reticulum because it forms a network in the cell. And if we need an electronic microscope to study the endoplasmic reticulum, we will see it has three parts: cisternae, tubules, and third one is vesicle. These are the three parts: cisternae, tubules, and vesicles. Now, you will see cisternae are just like these stack-like structures placed in piles, in heaps, just like this. And these include a space also. The space is usually filled by fluid, which is called endoplasmic matrix. Endoplasmic matrix is the fluid space, endoplasmic matrix. Its second part is tubules. These are extensions of the endoplasmic reticulum and are just like network, coiled form, just as this. These also have endoplasmic reticulum. And third part are vesicles, which are usually spherical and are filled by fluid. These can be isolated, or these can be attached with the tubules or the system. If you examine deeply the endoplasmic reticulum under an electron microscope, you will see that there are some cells where endoplasmic reticulum has small granules on its surface, just as this, and it looks rough with the presence of those granules. Actually, these granules are ribosomes, and these, in that case, the endoplasmic reticulum that provides a stage for the formation of proteins. Such an endoplasmic reticulum is called a rough endoplasmic reticulum. I will write rough ER or ER. Rough endoplasmic reticulum. Its surface looks rough, and this roughness is due to the presence of ribosomes on its surface, and ribosomes are protein factories. These help in the formation of proteins. These are very helpful, and this here endoplasmic reticulum, it will provide a stage for formation. So some of our organs like pancreas is there, liver is there. If we will examine endoplasmic reticulum there, because pancreas, liver, etc., they need higher quantity of protein. For that purpose, they need rough endoplasmic reticulum. Now, second one is where protein formation is very low, where the cells do not need, or organs do not need, so much type of protein, so much quantity of proteins. You will see that the endoplasmic reticulum will look smooth. That means there will be low quantity of ribosomes, or there will be no ribosomes. Those are called smooth endoplasmic reticulum, or SER, smooth endoplasmic reticulum. So two types of endoplasmic reticulum are usually present. It is actually same, but the type occurs due to the presence of ribosomes on its surface or no. The main function of endoplasmic reticulum, if we calculate, is maintaining the shape of the cell. It maintains the shape of the cell by by its network, and it also helps in the moving of molecules from outside inside or from inside outside. So it's a passage of different material from outside to inside the cells. It maintains the shape of the cell. So this was endoplasmic reticulum, one of the important organelles of the eukaryotic cells, and I will once again tell you that in prokaryotic cells, in bacterial cells, these are absent. Fungal cells have endoplasmic reticulum. Similarly, plants have endoplasmic reticulum. Animal eukaryotes have endoplasmic reticulum, but bacterial cells do not have endoplasmic reticulum.